The world record for wheat production is just shy of 18 tonnes a hectare. Today in this video, I'm going to share with you the 7 critical points of influence that we can have on our crop to achieve as much of that potential yield as possible. Now, it's important to note that was set in the UK, where there are certain environmental conditions that we don't get in Australia. However, hitting each of those critical points of influence on our wheat yield is very important in order for us to achieve maximum yield. Now, if you're more of a visual reader, we have all of this written up on our website, as well as the guide as to when you need to differential sap test and when to apply, as well as our recommended seed treatments and recommended products that we can supply our clients with. There's a link below to do that. Now, this stems from the understanding that when you have a seed, you start with the maximum yield potential. To say we have a graph where we have time on the bottom and we have yield at the top. As soon as we plant that seed, we start with the maximum genetic potential of that plant. Every time that plant experiences stress or a nutritional need that's not met, it steps down and down and down and down until we get to our final yield. So yield is not made, but simply lost. So whenever we're not applying the correct nutrition or water to our crop, we are losing yield. So that's super important, because the things that start down here at the start of our wheat's life will ultimately affect us down here at harvest. So today we'll be covering these 7 steps that I've identified as very important. I've mapped them to our growth stages. Some of this is an aggregation of lots of different research, primarily, or the main lot of research comes from John Kempf's critical points of influence. However, I've expanded onto that to add some more and then mapped it to wheat, as well as a few other papers. Now, the very first, very critical point of influence that we have on our crop is our seed selection and how we treat that seed. Now, there's a number of different cultural practices like the planting depth and timing that will affect everything else, but there's 4 factors that we really focus on. The first one is seed size. So that is having basically the biggest wheat seed you can get. With that, you're going to have an increase in reserves for that plant. Get up and out of the ground as soon as possible. So a larger seed doesn't necessarily mean you have a greater germination rate, but it does mean you'll have a stronger seedling to rapidly get out of the ground and start growing off the reserves from the seed, allowing for a really firm foundation for the rest of these other steps. So that's the first one. The second one is biology. Biology in both our ground and on the seed itself. So this is when we get into more or less the epigenetics, or thinking of seeds as its own inoculant, bringing its own microbes with it, which is why it's quite important to get your seeds from a really good supplier. I think there's a big opportunity in improving the environment in which our seeds are grown to bring all those microbes with it. So these are things like mycorrhizal fungi, making sure we have a really good mycorrhizal fungi population in our soil, or at the very least apply an inoculant. It's that mycorrhizal fungi, for example, that will assist with uptake of minerals that's important for everything else. So what this means for you, in terms of both these points, size and biology, getting our seeds as big as possible, so the largest test weight, and making sure it comes from potentially a regenerative farm or farms that are using biological approaches to enhance their production. Next in this is we're looking at applying seed treatments. Now we'll have a whole video coming out on seed treatments in the future. These are things like applying our microbes as well as nutrition, and not applying fungicides to our seeds to give them that package of nutrition and microbes to get out of the ground as soon as possible. Get the microbes right into that seed coat, so as soon as that root comes out, you are forming these symbiotic relationships with microbes, supplying it nutrition. This can really boost basically everything. This is the lowest hanging fruit for any farmer. If you're not applying seed coats, I'd highly recommend trialing it out. Again, we have a video coming out talking about the costs and benefits of a seed coat and how it would compare to a fungicide in both the actual performance, germination, and seedling development, as well as future protection from different diseases. So that'll be a super interesting video. Highly recommend watching it. Finally, genetics, making sure we have the right genetics to what we want our crop to do. If we're looking for a crop to perform in a lower input environment, finding genetics that will suit that. Likewise, if you have an environment where you want a longer growing season or shorter growing season, genetics, super important. These 4 things build the foundation for the rest of the crop. In terms of when this occurs, it technically occurs prior to the seeding of the seed. So it's actually the development of the seed from the previous crop, which is why the biology in size is so important, and to know where our seeds come from and the quality of that is important. But really it's between our growth stage 0 and 10, which is where all of this kind of sets the scene for everything else. So that's when our roots develop, and just before the first leaf comes out. So having that will set our yield potential as high as possible, and making sure that we can build on that and prevent any losses during that stage. Now the next stage is quite critical as well, and it determines the number of tillers per plant. It determines the spikelets per spike, as well as our grains per spikelet. And so if you think of yield for a wheat crop at least is determined by the number of tillers per square metre or per hectare, the number of spikelets per tiller, and then the number of grains per spikelet. So when we maximise each of those, that's how we really maximise yield. And now it's common belief that this is actually determined during the booting stage, but it's, although that's correct in the sense of it occurs during the booting stage, the head determination or the tiller determination and all the stem cells turning into the actual specialized cells happens very, very quickly after germination, within a few weeks. And so between the third leaf and basically the first tiller is when these cells differentiate. So it's super important that we have these minerals, all of which help with cell differentiation and cell division, sets the scene for this. So this is also why it's really important to have a really good seed quality, as it might be difficult to get a foliar application onto 3 leaves because there's really not much of an area there to hit. And this might be good to apply either in furrow as say a starter fertiliser, or include it on your seed treatment. So it can be difficult to foliar apply these, but there is a potential to apply these minerals towards the later stage of seedling development. However, it's really at that third leaf that these things start to happen. So as I said, each of these stimulate cell division or cell determination. So calcium is quite important in developing cell wall. Now manganese is also quite important as it regulates potassium. Now, a very important thing to understand is the way our minerals interact with each other. So calcium and boron are synergistic with each other. It means you really need boron in order for your plant to uptake calcium. You might have a ton of calcium or tons of calcium in your soil, but if you don't have boron, your plant can't pick it up, so it's required. Which is why in that critical point of influence, we need both calcium and boron. So making sure we have both of those. Calcium and potassium, they're both cations, they both use the same uptake strategy or pathways, which means if we have too much potassium, it's going to antagonize the uptake of calcium. Now, calcium is needed, especially in this stage, as well as many of our other stages, but we really want to be managing this calcium to potassium ratio in our plant, and we use differential status analysis to help guide that. But really, applying a lot of potassium up front can damage our yield potential into the future because it inhibits the uptake of calcium, which is important for cell division. Finally, manganese, which we just talked about, is important for regulation of potassium. So it increases and reduces potassium so that's in the required or ideal amount. Now, a lot of this is according to basically what I call the Bible of plant nutrition. This book is fantastic for understanding everything you need to know about the different minerals and how they interact with both each other and the plant. And a lot of my understanding has come from this book. Highly recommend getting it if you're a big nerd about crop nutrition. Otherwise, you can hire us as a consultant to do that for you. But the other minerals required, zinc. Zinc is also required in the formation of the cell wall to more or less an extent as calcium, but there are a few references to that. Copper is important for lignin production, also assisting in that cell wall integrity. Urea can be applied if it's necessary or needed, so this is a bit of a maybe one. Foliar application of urea is really efficient, it's really good. So we would only do this if it's paired with a differential status analysis, because we really want to know, do we actually need it? Because otherwise, excess nitrogen can cause a whole range of different problems. That can cause a yield penalty, when we don't manage it correctly. Now finally, kelp. Kelp is super cool. It's full of plant hormones called cytokinins. Cytokinins are quite important as it stimulates cell division and cell elongation. So in this context, it can assist with the differentiation of the stem cells and help to divide them up and to produce more cells. So if we hit this nail on the head, we'll maximize the amount of tillers that can potentially come out, the number of spikelets per tiller, and the number of grains per spikelet. Now it's important to know this doesn't guarantee that we're going to have heaps of tillers. It just means we have the potential to. And so the rest of the stages allow us to fill up our maximum to this. So say we hit the nail on the head, we maintain that yield potential. We're not stepping down because we missed a bit of calcium that didn't allow us to put on an extra tiller. Because no matter what we do for the rest of the season, if we're minimized or if we're limited by this stage, we can't do anything else. So as I said, we take a differential sap test at the top. So we could either do a differential sap test on basically the newest leaf or the first and the third leaf, which is a bit of a pain, or we just take a whole top sample. So we take the whole thing and just do a sap test on that. Super good enough for this stage. The important thing with trying to get these things right is that by the time you take your sample, send it to the lab, it's 7 days later. By the time we get the results, we can read the results in an hour or two. It's then getting the fertiliser or the nutrition you need to your farm. So sometimes you might have to guess and pre-order some of these things so that you can then make the application the day of you get the results back. Otherwise, if we try and guess these up front with our starter fertiliser, then that could be a potential way to overcome it. We use in our seed treatments a lot of these trace minerals so that we set the scene for this. This is a maybe. Maybe if you're really trying to push yield, you might do a foliar spray. But otherwise, we kind of consider this in our seed treatments. The next critical point of influence that I've identified is the photosynthetic ability of the plant. Now this basically goes from the end of the seedling development stage all the way up to pre-flowering. So it's a long stage, but it also sets the scene for everything else. The aim of this stage is to maximize the photosynthetic ability of the plant, especially that flag leaf. The flag leaf contributes, I think it's 40 to 50% of the carbohydrates in the grain. It's a massive amount of carbohydrates that basically just one leaf per tiller produces. Likewise in the stage, we want to maximize the amount of root exudates to feed our biology that will feed us later in the stage, and setting just a really healthy, good plant, quite important for carbohydrate production, which is needed later. So during this stage, we might take 1 to 3 sap tests or differential sap tests to guide our foliar applications. Now again, this all depends on what type of system you're running. If it's a high input, you're trying to maximize your yield, your irrigation going, you've got all these things going, and you're trying to get that 18, hopefully 20 tons a hectare if we can achieve it. That's a pipe dream, but could be possible. They might take 3 or 4 differential sap tests to do that. If you're more or less a lower input system, we might take 1 differential sap test to guide our nitrogen placement or our application. But anyways, to maximize our photosynthesis, we need a few things. The first is nitrogen, making sure we have sufficient amount of nitrogen for the chlorophyll molecule. Now we don't want to overdo nitrogen, and we want to make sure it's in the right form. So a foliar urea base is quite good as it's more efficient than anything else. We'll have another video coming out about nitrogen use efficiencies and the different forms and how we can maximize plant energy with the right form of nitrogen. So urea, otherwise amino acids are quite good. They're a little bit more expensive, but give the plant more carbon. Magnesium is important because it's in the center of the chlorophyll molecule. This captures the light, quite important. So on our differential sap test, we're looking for these things as well as our bricks to understand overall sugar. Manganese is important for the splitting of the water during hydrolysis, as well as a few other things. Iron is used in carotenoids, which capture a broader range of light. Phosphorus is also used in ATP production, which translocates, it's more like the energy storage or the batteries of the cell. We want to make sure we have heaps of these because when we have all this energy produced in photosynthesis, we want to be able to capture it somehow, and we need ATP with that. So making sure we have all of these is foundational to the driving force that produces the energy of the plant. Really want to make sure we're nailing this on the head, because from this we get everything else in the plant. We get all of our proteins come from this, all of our secondary plant metabolites, all our defense compounds, all stem from maximizing our photosynthetic ability. If you're interested in that and learning how that flows onto plant health, we have a video out called John Kempf's Plant Health Pyramid, where we deep dive into the nutrients required for that. Next is protein synthesis. We want to maximize the quality of our proteins that our plant produces. Now, proteins are quite important because they provide basically a whole range of different functions in the plant. If you think of photosynthesis as producing the feedstock, it's the proteins that we want to produce for plant defense, as well as any other function of the plant. So for that, we need the nitrogen, but assuming that we have the nitrogen already, what we need is sulfur. Sulfur is required in the development of two amino acids. So that's methionine and cysteine. Now, methionine is quite important because it codes the first amino acid for every protein, which requires sulfur. So if you don't have sulfur, you don't get any proteins. It's quite important that we have a ratio of 10:1 for our sulfur. So our nitrogen at 10 parts and our sulfur at 1 part. That's going to ensure we have all the sulfur required for our amino acids to be produced. Magnesium is required in the ribosome, which puts together our amino acids into peptides and polypeptide chains. Boron is required for the translocation of the proteins or nitrogen and everything around the plant. So it's not so much used in protein synthesis itself, but more so for the translocation of nitrogen around the plant, as well as everything else like the carbohydrates, because it's in this stage that makes us prone to fungal and any other disease attacks. So we want to make sure we have complete proteins, as fungi and other pathogens don't find complete proteins as a valid food source. Now, this is very important when we consider our flag leaf and making sure our flag leaf is in a really good nick, as again, the flag leaf contributes so much carbohydrates to the plant. So making sure it's in good nick, good health to defend against or not be a food source for pathogens, which is why we're focusing on this protein synthesis stage. Lastly, we have molybdenum and nickel. They're important for molybdenum. It's the nitrate reductase enzyme, so it converts nitrate into nitrite for then to be processed into amino acids. Likewise with ammonium, nickel's important for that conversion. Finally, we want to keep this stage in more or less a reproductive stage, making sure we get a really nice vegetative growth, because it increases leaf size and the amount of photosynthesis that can occur. So there's 3 minerals that can really stimulate that. That's calcium. Calcium is a really good quality driver of vegetative growth. For that, we also want that boron. Silicon is also important for vegetative growth. It builds a really high quality plant. Also increases the plant tolerance to any biotic or abiotic stress factors. And another mineral that I didn't mention here, but it's zinc. Zinc governs leaf size. So making sure we have enough zinc, making sure we've got a really wide, healthy plant leaf will also maximize our photosynthetic ability. So all of this, making sure we maximize photosynthetic ability with a really healthy vegetative energy and production, and making sure it's of high quality so that we're not getting pest and disease attacks. We'll make sure we have a really good plant with strong roots, a good driver of photosynthetic ability to then fill our grain with carbohydrates. So the next stage that we can maximize our yield is the flowering stage. Now, there's really 2 points we want to hit on this. It's pollination success and grain set. So only 35 to 40% of the flowers or of the florets on our spike or our grain head is actually fertilized and therefore turns into grain. When they're not fertilized, they abort and they fall off, and so we don't actually get that as grain. So this kind of proves the point of why it's important that each step is met, because if we don't really hit this on the head, or hit this on the head, we're still limited by this stage. Likewise, hit this nail on the head, but we have a poor flowering, then we really lose our yield during that stage. So with this, we want to make sure we have a really robust pollen. So for that, we need calcium, manganese, boron. Again, that regulates potassium. This regulates the cell wall, or the strength of the wall. Boron regulates or increases the uptake of calcium. Zinc is important. Copper is quite important as it governs the fertility of the pollen. It also increases the lignin production in the anther, so making sure we have basically a really strong flower on our plant. Kelp is also really important, something we can apply. It helps with the cell division and cell elongation, so by providing cytokines. And phosphorus is important for flowering. But yeah, we kind of know that. But given that many agricultural soils are high in phosphorus anyways, this is why it's lower on the list. Calcium is typically not highly available in our plants, at least when we look at subtests. So calcium, very important for this, as well as copper. Copper can be a bit of a pain to apply as a foliar. It doesn't really mix well with other things, so that can be a bit of a pain, but potentially worth it depending on your operation. Now with this, and with each of these, typically we like to apply a foliar spray. With that, and trying to hit these points of influence, we want to make sure that we're actually applying the foliar spray a week to 2 weeks, or 2 weeks to a week prior to the actual event occurring. So there's no point in applying foliar spray right as it flowers at the Z60 stage, because everything's set in stone. Everything's set in stone for the plant to already flower. We want to hit it prior to it flowering so that it can actually use those nutrients in pollen production. So, which means if we're going to apply, say, a week beforehand, we need to take our differential subtest 2 weeks beforehand so that we can then hit at the right time. So now we have one of the most important components of the critical points of influence, and this is cell division. So once the embryos are fertilized, as soon as that happens, the cell starts to go from one cell, which is the embryo, and starts to divide into many cells. And so we get this doubling effect. Effectively, during this stage, we want as many doublings as we can get, as many cells that can be produced, because it's these cells that later get filled up into our grain. So this sets the density of cells within our grain, and it's very, very important. So as I said, number of cells produced, it occurs immediately after fertilization up till 10 to 14 days. So say 10 days. So Z60 or 65 is midpoint flowering. So taking our self-tests more or less at flowering will give us a good understanding of what we need as soon as flowering ends. So with this, we really want to manage potassium. Potassium, again, is antagonistic to calcium. Calcium is very important for cell division, which is what we want to drive. So making sure we have a really good amount of manganese to manage potassium. It's not so much for manganese itself, but more so to manage potassium. And then again, we want calcium and boron for that quality cell walls. Really help with cell division. Now, I have suggested kelp here as well because of those cytokines, which help with cell division. Kelp does contain, or at least the product we recommend, is 1% calcium, 1% potassium. So there is a little bit of potassium in it. So just be careful. If you have too much potassium, you might not want to apply it. But it contains those cytokines, which really help drive the system. So if you're slightly low on the potassium, it's not a bad idea to add it because it is balanced off with the calcium. But overall, would highly recommend that in that foliar application. So next is grain fill. Grain fill is the other critical point. If you're, say, a very low input farmer and you only can choose 2 of these, I would choose your seed. So seed treatment, greatest return on investment, and your grain fill in terms of your critical points of influence you want to hit. So this component is filling up these cells that we just produced with carbohydrates and potassium and proteins, or more or less. That's really what we're filling up the cell with. And so we want to maximize the amount of stuff we put into those cells because that's where we increase our yield because we're putting on all that weight per grain. Now the minerals for this actually switch. Potassium becomes very important, which means we want to minimize our calcium. So basically we have this stage where we don't really want much potassium and then we're applying a ton of potassium. So again, we can do that with a foliar application. We recommend a really good product that's full of potassium. So hit that potassium here. Potassium helps with the translocation of carbohydrates as well as itself into the cell. So it really loads up the grain with carbohydrates, which is where we get that yield from. So after that, after potassium, we want to make sure we hit nitrogen as well, hitting our protein requirements in our wheat. Amino acid base is best. I'll have a video on that, as I mentioned before, but urea is also a good option. We want to make sure we add a carbon component with that, so the plant doesn't then have to take carbohydrates from somewhere else and then put it with our nitrogen. So next we want manganese. Manganese again for the benefit it brings to potassium. Sometimes the low potassium in your plant might not be because of low potassium itself, but the lack of manganese. So we want to make sure we have that regulator for the potassium. Zinc is important. Boron, which facilitates the uptake of your carbohydrates and everything as well into your grain. Calcium is important, copper's important, and then kelp as well. So kelp with the cytokines increase or stimulate cell elongation, which effectively is this. We want to have a really big cell full of carbohydrates and protein if that's what we're going for. Now, finally, our final critical point of influence is ripening. Now, ripening, we can influence to make it go longer or shorter depending on our context. So the first two minerals, we can either apply boron to shorten ripening, so allowing us to harvest quicker or sooner. So this increases the amount of carbohydrates that get pumped into the grain, at which point, once the grain starts to fill and mature, it releases ethylene. Ethylene then causes the senescence of the whole plant. So it causes the plant to die. So boron makes that speed a lot faster, a lot more rapid. However, on the other hand, cobalt actually suppresses that. So cobalt can actually delay maturity. It increases the production of cytokines as well, which suppresses ethylene. So we're not getting that senescence. And we can extend basically this ripening stage of the grain fill stage by a few days to a week, which means we're still photosynthesizing, we're still pumping carbohydrates into the grain. That's how we're maximizing our yield and therefore money. Finally, we have our protein synthesis compounds. We have magnesium, sulfur, and molybdenum, potentially nickel, but less so. And this is important to convert any nitrogen into proteins that we want in our grain. And then we can harvest our 18 tons a hectare wheat and get a metal. So depending on your farm and your operation, I would pick the seed coat as the most important thing you can do. Maximizes your return on investment. This is super important because it flows onto everything else. This flows onto your head determination, photosynthetic ability, flowering, cell division, senescence, and grain fill. So if you're a farmer and you really want to have a low input system, applying the biology onto your seed coat as well as more or less starter nutrition onto your seed coat really pays massive dividends later in the season. Now, I touch on biology. Biology, especially as you'll notice during here, calcium comes up a lot. Calcium is delivered in a fair amount with the rhizophagy cycle, as well as other symbiotic relationships. Mycorrhizal fungi also deliver zinc, which comes up a few times, phosphorus as well as water and any other trace minerals. So making sure we have the biology on our seed coat and making sure we have a really good population of microbes in our soil can really help deliver nutrients at different points depending on what the plant needs. Likewise, we prefer foliar applications guided by differential subtest analysis. Gives us the best understanding of what's actually going on and we can really target our applications. Otherwise, that's really it. They are the 7 critical points of influence and allow us to set a high yield potential and maintain that by the time we harvest. If you like any of that, we offer our consulting services to help you achieve these kind of things, regardless of the enterprise you're running on your farm. This works in broad acre as well as horticulture or perennial as well as small crops. More or less to pasture, we're really looking at photosynthetic ability and vegetative growth. But overall, if this interests you, interested in maximising your yield, but not just maximising yield, but maximising profit, as oftentimes you can use less fertiliser, but at the right time to maximise your yield. So if that's of interest, sign up for a free consultation on our website at agresol.com.au. My name's Till Simmons from Agresol. Thanks for watching. Cheers.